Hi, guys. So we are going to be, we just finished this part, right? Yeah. We're about to do here. Yeah? Okay. So here's my request of you today. Cardiovascular system is logical but complicated. So you have to be honest with yourself and me about when you get confused. You have to stop me because all of this builds on itself, right? So some of you guys are extroverts and have no problem stopping me, but I'm also telling you that the introverts need to raise your hand or at least poke somebody that will raise their hand for you, right? Whatever, because this is all just building relationships between things. As far as expectations, remember we're moving your test to a week from today instead of Tuesday, right? And what you are going to have over the weekend is a take-home collaborative quiz. I'm going to try to make it kind of complicated so that you have to talk to each other about it, okay? And I'm also going to make it cover the things that we haven't covered yet on Tuesday. So I'm not going to close it on Sunday. I'm going to close it on Tuesday, right? So if you got stuff wrong, just like we did last time, you'll have time to fix it after you listen to the lecture. Okay, good. But I also want you to work on it over the weekend so that you can start to figure out what you know and you don't know, okay? So cardiovascular system, you kind of need to practice it. It's a bunch of little puzzles. Go ahead.
Is the one before graded? Because it says currently open on my thing.
Oh. I don't know why. It's just, I probably just put in the wrong date. It's graded. I'll just close it. I just probably put in the wrong date for closing it. Okay, but you can go in and change it again if you want. I don't care. If you got it right now, cool. Okay, so cardiac muscle, what we were doing last time. So remind me of a couple of things before we get going. First, what is the overall physiological function, the trunk function of the cardiovascular system? Blood flow to capillaries. Exactly. Keep that in mind always, okay? And then remember that last time we basically said that if we serve the god of flow, right, the two primary considerations for flow are delta P. Somebody explain to me what delta P is in your words. What is delta P? What? It's not the change in pressure. It's the what?
It's like the pressure difference.
Pressure difference between 2 points, yes? Okay? Okay. And pressure difference between 2 points. And so if pressure difference increases, flow increases. And somebody explain to me in your words what resistance is, the R. What is it?
The interference of the flow. So
yes.
An example of that is longer arteries.
Yeah, longer tube, narrower tubes, whatever, those kinds of things. More viscous fluid. This is the opposition to flow given a standard delta P. Just hold delta P constant and say, okay, what could impact flow between these 2 points? So what we are going to do today is we're going to be talking a decent amount about pressure. Do you remember that there's 3 ways to manipulate pressure that we talked about last time? What are the 3 ways? Yeah, what your vessels do, constriction and dilation. Yeah, what your fluid. Yeah, how much fluid volume, and then what else? Pump. Yeah, heart, vessels, fluid. Yeah, heart, vessels, fluid. So what we're going to do today is we're going to talk about the ways to manipulate pressure. But first, what we need to do is go back and pick up some action potentials and contractions in cardiac muscle. It's not going to be super hard because you've done variations of this before. So what we are going to do is we're going to start talking about cardiac muscle tissue, and we're going to go back and talk about the pacemaker cells in cardiac muscles, yeah, and then the contractile cells in cardiac muscle, and look at how they behave and cause contraction. Before we get to that, we didn't talk about this yet. Did we talk about ANP or ANF? Okay, so just a couple of little tidbits before we get into how the cells actually work. First, cardiac muscle cells, some of them have the capacity to release a hormone. And it's a hormone that you haven't learned yet, but you can add it to your endocrine list just for patho or whatever, and it'll all be in one place. This is a hormone that is released from cardiac muscle cells called ANP or ANF. Listen to the name and see if you can figure out something from it. Atrial, which is where it's coming from. Natriuretic implies but doesn't tell you the target. What target does it imply? Kidneys, yes. Atrial natriuretic peptide or factor, F or P, nobody can agree. Okay, so you guys, I'm going to tell you why it's released, and you're going to try to see if you can figure out what it does. And yes, Anna, think negative feedback. Okay, ready? It is released in response to prolonged atrial wall stretching, and it targets the kidney to do what to blood pressure? Decrease blood pressure. So is it going to make you pee or make you not pee?
Pee.
Make you pee. Yeah, it's going to make you pee. Good? Okay. It's not released just if your atrium is stretched like when you're exercising. Yeah? It's released in response to prolonged atrial wall stretching. Good? Okay. Okay, the other thing before we get into cardiac muscle cell action potentials and stuff is just the metabolism of cardiac muscle. Okay. Cardiac muscle is not at all picky about nutrients. So it can, for instance, catabolize glucose and other sugars. It can also catabolize fatty acids and lactic acid and all that kind of stuff. So getting in nutrients to cardiac muscle is not generally a problem, but it has almost zero capacity to do anaerobic cellular respiration, which means oxygen is the problem. So what happens if you don't get adequate oxygen to cardiac muscle is what's called intolerant of ischemia. And ischemia is reduced blood flow. And so, of course, you're going to have pain. That's like angina. But you can also have a heart attack. And a heart attack is when the cells die because of lack of oxygen. And that's especially problematic with cardiac muscle because what can you tell me about cardiac muscle's possibility of mitosis? Yeah, pretty much zero, right? Cardiac muscle has almost zero capacity. So whatever damage occurs and kills your cardiac muscle cells doesn't just create a momentary problem. It creates a long-term problem because you ain't getting them back, right? So that makes sense? So oxygen is the primary consideration here. Okay, so now we are going to get to cardiac muscle cells. We'll deal with this in just a second. Okay, so cardiac muscle, where are you? Okay, two different categories of cells in cardiac muscle. There are what we call pacemaker cells, which are concentrated but not exclusively just concentrated. They're in the SA node and the AV node. I'll show them to you in just a second. And we need to figure out how those work. And then there are also contractile cells. Contractile cells are the ones that are going to contract, and they're the ones that are striated, and you usually think about when you look at cardiac muscle tissue. Okay, so we've got two different categories of tissue, or categories of cells in the tissue. So about 1% of your cardiac muscle cells are going to be those pacemaker cells. A lot of times they call them a bunch of different things. They call them pacemaker cells. They also sometimes call them autorhythmic cells, yeah, or nodal cells, because that's what they are. I'm going to try to call them pacemaker cells because that's pretty consistent. What happens during embryonic development is pacemaker cells lose their ability to contract, so they're just specialized for spontaneous depolarization with gap junctions to pass on the ions to their neighbors. So they spontaneously depolarize and pass it on. So sometimes they're called conducting cells because they spontaneously polarize and pass it on, spontaneously polarize and pass it on. They are conducting action potentials on their neighbors, right? So these are pacemaker cells. Pacemaker cells, remember from anatomy, are concentrated in a few places. They're also kind of scattered around. But SA node, do you guys remember where the SA node is? Yeah, in the wall of the right atrium, just inferior to the opening of the superior vena cava, right? AV node, inferior part of the interatrial septum. Do you remember the rest of the electrical parts? What's this part? No, that's down here. This is the bundle of His or the AV bundle, right? And then you split into the left and right bundle branches and then the Purkinje fibers, right? Okay, so all of this that you're seeing in yellow here is the cells called pacemaker cells. It's just series of pacemaker cells, okay? Sometimes they're clumped up in a node and sometimes they're put one after the other in a chain like an AV bundle. Everybody with me? Okay, so this looks like nervous tissue, but it's not. It's pacemaker cells. Okay, so with these pacemaker cells, by the way, for those of you who ever use interactive physiology, it's fantastic for the cardiovascular system. It's super, super good. And a lot of times it's really helpful because you like have to manipulate things like increase the resistance, watch what happens to the flow. Decrease the resistance, watch what happens to the flow. So it's really good for these animations as well. All right, so the process of generating an action potential in a pacemaker, a cardiac muscle cell. It's weird. These cells, these pacemaker cells, what they do is they spontaneously depolarize, get an action potential, pass on all of the ions to their neighbors, repolarize, do it over again. So they are constantly just depolarizing, repolarizing, depolarizing, repolarizing. And they don't need the nervous system to do it. They spontaneously do it. And that's totally different than skeletal muscle, right? Skeletal muscle doesn't do a damn thing unless the nervous system tells it what to do, right? But these cells will spontaneously depolarize. It works well enough that you can, for instance, take a heart out and it'll still beat, right? Till it runs out of stuff, right? It's going to beat. So these pacemaker cardiac muscle cells, their action potential looks very much like this. Spontaneously depolarizes, hits an action potential, repolarizes, starts the whole damn thing over again. This is without attaching the nervous system, no hormonal activity to it. It just does this. How's this work? So let's look. The way it works is they have what's called a variable resting membrane potential, meaning they don't stay there for very long. They don't like just sit at rest. And what happens is they have a dependable number of what we call sodium funny channels. They used to call them sodium leak channels, but they call them, I don't know why they're funny. It's not funny to me, but they're like open under almost all circumstances. There is a way to close them. So they're not exactly gated and they're not exactly leak. So what the hell are they? Funny, not funny. So sodium funny channels. And what those do is, as long as you have a dependable number of sodium funny channels and a dependable sodium electrochemical gradient, which you worked really hard on, right? Then what's going to happen is you're going to have a dependable depolarization rate, yeah? And so you are going to depolarize to threshold, yeah? And threshold in cardiac muscle cells is minus 40, minus 50-ish. And hit the depolarization. At threshold, you open a new kind of channel, and the new kind of channel is going to be a depolarizing channel. So does it surprise anybody that it is a voltage-gated sodium channel? Oh, sorry, voltage-gated calcium channel? Can't you use calcium for depolarization? Yeah, right? So this is a sodium funny channel. This is a voltage-gated calcium channel that causes a depolarization. And calcium will depolarize simply because it's positive and wants to move in, right? Okay, calcium is going to rush in, okay? And then what's going to happen is I'm going to have to repolarize, right? So I'm not going to tell you guys how to repolarize. I'm going to let you tell me in just a second. But first, let's fill this in on this figure on your notes. You ready? Okay, so before we get to this portion right here, somebody tell me how I'm going to get from here to here. Open or close? They're open still. They're always open, right? And they are sodium funny channels, yeah? Okay, which are kind of like leak channels. They're not voltage gated yet. No. But here at threshold, we are going to open a new kind of channel. And what is it going to be? Be more specific. Voltage gated calcium channel. And I want you to write fast voltage gated calcium channel because we're going to meet a slow one in a few minutes. Fast voltage gated calcium channel. Yeah? Everybody with me? Okay, the fast voltage gated calcium channel. And then what has to happen is I am going to flood this with ions. Most of the ions I'm going to pass on to my neighbors, yes? Okay, I'm going to pass on to my neighbors through gap junctions because these are connected to their neighbors through gap junctions. But whatever I didn't pass on to my neighbors, I'm going to have to fix before I can depolarize again, right? So let's talk about how you would repolarize. So how would you repolarize? You guys tell me. Up here at the top of the action potential on these pacemaker cells, what do I need to close? What do I need to open? Close what? Close voltage-gated calcium channels, yeah? And then what? Open voltage-gated potassium channels, yes? And that would allow me to repolarize, right? Okay, everybody good? Go ahead.
We could also have a calcium pump.
Yeah, we're going to do that in just a second. And then we're going to have to pump everything because, okay, we've replaced the number, but everything's in the wrong place, and we can't do another one until we replace the location. So you guys tell me, what kind of pumps do I need? I need a calcium pump on the cell membrane, yeah, and pump calcium back outside, right? And then what other kind of pump do I need? I need a sodium potassium pump, and I'm going to pump sodium which way? Out, potassium in, yes? Okay, does that make sense? So do you see why, because you spent all of that freaking work understanding membrane potentials and action potentials, nobody has to tell you how to repolarize anymore. You're like, put everything back where you got it, okay? Do that. That's how you repolarize. So with this, again, at the top, I'm going to close calcium, open potassium, right? And it's going to allow for repolarization, and then you're going to need two different kinds of pumps. You're going to need a sodium-potassium pump to put the sodium and potassium back in the same place, and you're going to need a calcium pump as well. Everybody okay? Okay. So note to self, contraction doesn't follow in these cells. And you don't need to do as much pumping as you imagine, because most of the ions you actually sent on to their neighbors, right? So you flooded yourself, you sent most of the ions onto your neighbors, but whatever you had left, you have to put back in position, okay? So now let's talk about, so just conceptually, I want you guys to talk about, so this is going to do this inherently 100 times per minute because that's how quickly the SA node depolarizes, okay? But your heart rate is not 100 times per minute at rest unless you got something going on, okay? It's, you know, anywhere between 60 and 80. Should hopefully be a little lower. We'll talk about why later. So I want to talk about what the sympathetic nervous system does to this and the parasympathetic nervous system does to this, because this directly correlates to what we did last time, okay? So stick with me. If I am sitting here on this picture and I am inherently depolarizing at the SA node 100 times per minute, but then I activate the sympathetic nervous system, stimulate the SA node, the sympathetic nervous system is going to release what neurotransmitter? Sympathetic nervous system, norepinephrine. And what norepinephrine is going to do is I was on my way to depolarization, but I got there faster, yes? So more frequent depolarization, yes? Okay, so I was already on my way, right? And then I released something that said depolarize. And it was like, I was already doing it. It's like your kids. They were taking out the garbage and you tell them on the way out, take out the garbage. And I'm like, oh. So it was on, it was, because then you don't get credit. So you were on your way to depolarization right here, but instead of waiting, you actually hit it right there, and then right there, and then right there, and then right there, yeah? So it increases the frequency of depolarization. Does it start or stop depolarization? No. As a matter of fact, if this isn't working, you can't speed it up. It's basically like hitting the gas pedal without turning on the damn car, okay? So the sympathetic nervous system can increase the speed of depolarization if it's already going. If it's not going, it's not going to do anything. What about the parasympathetic nervous system? Parasympathetic nervous system goes to the SA node as well, and it releases what neurotransmitter? Acetylcholine. What acetylcholine does is tries to hyperpolarize. So you were on your way to an action potential and you're almost there, and it says, no, slow down, right? So what would happen there is you would get fewer action potentials over the same time period. Does that make sense? Okay, I'll show you an image of that. Just give me a second. There's an image in your textbook that might help with it. I can't remember where it is. 13. Just wait right there. Where are you? There it is. Damn it. Balls. Can't find it. Kind of demonstrated it anyway. Huh. I don't know where it is. Huh? What'd you say? Was it the last one? Oh, it's this one. Okay. So this is what we were just talking about. Okay, it's busy, but start with at rest, the gray one. Okay, that's normal. Okay, that's 100 times per minute. Then apply norepinephrine, and then you get the green one, right? Okay, right. Start at the gray one, and then apply acetylcholine, and then you get the blue one. Does that make sense? It just slows down the rate at which you hit your threshold, right? With acetylcholine or norepinephrine. Everybody good? Okay. So now what we haven't even contracted. All we did was depolarize, and now we need to pass those ions onto the contractile cells and cause them to contract. But the contraction is going to be relatively easy for you guys to understand because it's really, really similar to skeletal muscle contraction. It's just got a couple little tweaks to it, okay? So the mechanism of contraction in the contractile muscle cells, so let's look at how that works. All right, so this figure should look kind of familiar to you. It's really similar to the skeletal muscle one, right? Only it doesn't start with the whole motor end plate, because how do I get my ions? I get them through the gap junctions, right? I get them through the gap junctions. But notice it's going to have the same thing. You start an action potential, goes down the T-tubules, causes calcium release, binds to troponin, all of that business. So let's go through it, okay? So how is this similar and different to skeletal muscle contraction? So the depolarizing stimulus, instead of getting it from the extracellular fluid, I get it from my neighbor, right? And my neighbor is a pacemaker cell. Maybe. I mean, you could get it from another contractile cell because they talk to each other as well. But let's say originally you get it from a pacemaker cell, right? And which ions are you going to be moving through those gap junctions? Calcium and sodium, both. Calcium and sodium. And they're both depolarizing, right? Calcium and sodium are both depolarizing. So you're going to move those through those gap junctions. And then if you hit threshold, yes, if you hit threshold, you are going to start an action potential, right? If you depolarize enough to hit threshold, you're going to start an action potential. I'll tell you about this guy in just a second. If you start an action potential, what you do is exactly what you'd expect to do. It goes down the sarcolemma and then down T-tubules and then causes calcium release from the where? SR. And then calcium goes and finds what protein to bind to? Troponin, and then moves tropomyosin. And then you cross-bridge formation, you do cross-bridge cycling as long as calcium is present. Yes? You do, and ATP. Of course, you need ATP as well. Calcium and ATP are present, right? So it's really the same kind of story that you had there, only a couple of little differences. So as the action potential goes across the cell membrane, it will cause the opening of another ion channel. And that ion channel is that little guy right there. That one is a slow voltage-gated calcium channel because it's not the one that initiates the action potential, but it's actually going to prolong the action potential. So I want you guys to think, if I let more calcium in from the extracellular fluid, am I going to have a stronger or weaker contraction? Stronger contraction. Is it going to be a longer or shorter contraction? Longer. Okay, so what's going to happen is this is going to prolong and increase the strength of contraction for each action potential. It's not going to add action potentials. It's going to do one action potential and it's going to be longer. So that calcium, does the troponin care where the calcium came from? It doesn't care, right? It's like calcium is calcium. It binds to troponin. It causes, more cross-bridge. So so what this is going to look like is you are going to get a really weird looking action potential. Okay, so what's going to happen is you're pretty much going to go from 0 to threshold almost instantaneously because you get flooded with ions from your neighbor. And when you go from 0 to threshold, you open this channel right here that is the initiation of the action potential. So let's look at that one. So what's going to happen is the depolarizing stimulus is going to cause voltage-gated sodium channels to open. This is a fast voltage-gated sodium channel, okay? And it's going to cause a massively quick depolarization. Then it's going to try to repolarize, but when you try to repolarize, now you're going to open a slow voltage-gated calcium channel, yeah? And prolong the action potential and increase the amount of calcium that's coming in from the extracellular fluid, okay? So when you do that, so the action potential was initiated by a voltage-gated sodium channel, prolonged by a voltage-gated calcium channel, right? And so I'm going to have a long, strong action potential because do I want my cardiac muscle cells to do weak little wimpy contractions? No, I want for every time I stimulate them for them to do a strong contraction, okay? So you can get strong and stronger. You don't do weak, okay? So what I'm going to have here is the action potential on a contractile cardiac muscle cell is super duper easy to tell from the action potential on a pacemaker cell, right? It looks totally different than the action potential on a pacemaker cell. So what I want to do in your notes, if you haven't already done it, is make sure that you get this labeled correctly. So I want you to tell me where to label the fast voltage-gated sodium channel. That huge upsweep from 0 all the way past, from negative 90 all the way past 0. Yeah? Okay. That's a fast voltage gated sodium channel, right? Then it tries to repolarize a little bit, but then you open what channel here? Slow voltage gated calcium channel, yeah? But now, again, I'm not going to tell you guys how to repolarize. I want you to tell me, okay? So how are you going to repolarize right here? Okay, what do you need to close? Close calcium and sodium channels, yes? And then what? Open potassium channels. That replaces the number, and now I have to put everything back where I got it. So I need 1, 2, 3 pumps, yeah? What kind of pumps do I need, guys? I'm going to need 2 calcium pumps. What kind of calcium pumps? Where are they going to be? I'm going to need 1 to go back into the SR, yeah? I'm going to need 1 to go back to the extracellular fluid. So here's your calcium pump back into the SR, back to your extracellular fluid. And then you're going to need a sodium-potassium pump, yeah? This one also shows you a new kind of pump that's a sodium-calcium pump. I won't hold you guys responsible for that one because you also have like 1 more. Okay, so again, I'm going to pump calcium 2 different places. Where? SR, yeah, and then ECF, okay. And then I'm going to pump sodium which direction? ECF. Pump potassium into the cell, right? And then everything's back into place where it goes. Does that make sense? Okay, all right. So let's just clarify, okay? So you have different channels used in pacemaker and contractile cells, yes? So here's your challenge, both with your take-home quiz and also your test, which will be a week from today. You have to pay attention to which cell I'm asking you about, right? When I say contractile, brings up different channels than pacemaker. So let's go through pacemaker again and make sure that we clarify what kind of channels are involved in pacemaker cells. So see if you can, A, of course, recognize pacemaker cells from a graph, right? Okay, those are pacemaker cells. Okay. B, tell me the channels that are involved in pacemaker cells. Before those, sodium funny channels, and then fast voltage gated calcium. Okay, that's the ones that are involved in that. But they both have potassium, so we don't need to talk about this. Okay, and now I'm going to do contractile cells. Okay, the contractile cells involve different channels. Yes. Okay. Contractile cells over here involve fast voltage gated sodium and then slow voltage gated calcium. Yes, slow voltage gated calcium. That's why these over here were called fast calcium, because these were slow comparatively. Yes, go ahead.
So there's nothing, so the figure 4 up there,
Uh-huh.
is there nothing happening there?
No, it just goes almost instantaneously to threshold because it gets flooded so quickly with ions. So there's not really like a much measurable upsweep. Everybody okay with that? Okay. Questions? Okay. So go ahead.
So we're also having NAK pumps there.
Yeah, we totally have sodium potassium pumps on both of them.
And that would be like at the end of the report.
Yeah. I mean, it's beginning, middle, and end, but definitely at the end, for sure, yeah. Okay, so let's talk about, do you ever want tetanic contraction, summation and tetanic contraction in cardiac muscle? No. You want every action potential to cause a strong contraction. You don't want to have to build up action potentials to get a strong contraction, because tetanic contraction in cardiac muscle will stop the pumping action of the heart, right? It goes like that. So with that, one of the ways that the cardiac muscle prevents tetanic contraction and summation is by controlling the absolute refractory period. Do you remember absolute refractory period is the period of time when you had an action potential and you can't get another one, right? In skeletal muscle, the absolute refractory period is 1 to 2 milliseconds, and then you can get another action potential and more calcium release, and you can get summation. In cardiac muscle cell, you do not want that to occur. So those voltage-gated channels do not reset for 250 milliseconds. So it's a long, forced absolute refractory period. Do you remember the absolute refractory period is really the resetting of the voltage-gated channels, right? Okay. So specifically the voltage-gated depolarizing channels, sodium or calcium or whatever. So what happens with cardiac muscle is those take a really, really long time to reset. So the cell is really done with its contraction before you can have another action potential. Does that make sense? And that forced long absolute refractory period means that you don't get any tetanic contraction in cardiac muscle, right, under normal physiological conditions. Now, we know that people can go into ventricular fibrillation and have all kinds of weird shit happen, but we're talking about normal physiological conditions. And why is that so important? Well, it's so important because, of course, it's just as important that your heart relax as it is that it contracts, yes? Okay, and for a couple of reasons, and I know intuitively you know this, but let's just work through it anyway. Okay, so of course, cardiac muscle and ventricular specifically contraction forces blood into the arteries and ultimately to the capillaries, and we know that that's really important. But cardiac muscle relaxation is what allows for filling, yes, which allows you to do that. And if you get it, is there any such thing as too fast a heart rate? Totally. If your heart rate goes too fast, it's not going to fill and you can't pump, right? So there's like a sweet spot for heart rate, and it can definitely get too fast. But there's another thing that it took me an embarrassingly long time to understand, and that is that ventricular relaxation is super important for filling, and we all get that, right? But ventricular relaxation is also really important for delivering blood flow to the heart muscle itself. So I want to like work you guys through this. Okay, ready? So the heart is, of course, a systemic organ, and it needs oxygenated blood. Everybody with me? Okay, so here's what happens when the ventricles contract. Specifically, we're looking at the left ventricle because we're trying to deliver systemic blood, right? So the left ventricle contracts and it pushes blood into the aorta, right? And the aorta is, of course, delivering all systemic arterial blood to everywhere, right? Okay, the first two branches of the aorta go to the heart itself. What are those two branches? Right and left coronary arteries. Okay, you with me? Okay, so here's what happened. The left ventricle contracts, pushes it into the ascending aorta and into the right and left coronary artery. But when it contracts, it also compresses the capillaries of the heart. It's squishing the capillaries of the heart, yeah? So it's like squishing a sponge underwater. Is it actually taking up the water? No, it takes up the water when you relax your hand. So one of the other things that happens if your heart rate is too fast or if you don't let your ventricles relax, like with ventricular fibrillation, is you squish the ventricles so hard that you actually cut off blood flow to the muscle of the heart. So it's just as important to relax the heart for filling as it is to relax the heart so that you can actually get blood to the capillaries of the heart. So it seems counterintuitive, doesn't it, that your heart beating really fast would actually cause a heart attack? That's why. You're cutting off blood flow to the ventricular myocardium. Isn't that kind of interesting? And it took me just way too damn long to understand that, right? You guys were probably like, I knew that yesterday. Well, shut up. It's probably because I taught you anatomy. That's probably why you know it. Because I didn't. I didn't know it when I took it. Okay, so you got the whole refractory period kind of thing? Okay. All right, so now what we are going to do is that's just the cells, and we're going to go back to this story that we were telling, right? The F equals delta P over R. And of course, the primary function of the cells that we were talking about is really just generating the P. Yeah, that's what they're doing. Okay, so what we are going to do is we're going to look at this. If you don't like it, there's tons of other ways to look at this, but it's a good graphic representation. So what we said is that blood flow to capillary beds is the god that the cardiovascular system serves, yes? Okay, and we basically said that blood flow is an interaction between pressure, positively correlates with blood flow, and resistance, which negatively correlates with blood flow. Yes? You with me? Okay. And then we also said that there are 3 ways to manipulate pressure. Yeah? What your heart does, which is about what we're about to talk about. That's called cardiac output. Okay. Blood volume. That's going to be primarily related to your kidneys. And then what your vessels do, which is like manipulating the size of the vessels. Okay. So what we are going to do right now is we're going to try to figure out how you can manipulate pressure specifically with the heart. Okay. And that concept is called cardiac output. Okay, so it should make sense to you that what your heart does directly changes this pressure, right? Okay, so let's look at that. So the concept of cardiac output is really pretty easy to understand. Cardiac output is simply the amount of blood that is ejected from your ventricles per unit time, and it's usually measured in some unit of volume, like a liter, for some unit of time, like a minute. I mean, you could do milliliters per second if you wanted to, but liters per minute. So, and it's really super, super simple logic, because cardiac output is simply the amount of blood that leaves your ventricles, and specifically, by the way, we're always talking about systemic circulation unless I specifically tell you that we're talking about the right ventricle. So we're kind of going to focus on systemic circulation. So cardiac output, the amount of blood that is leaving per unit time, is simply a factor of how many times you beat per unit time, right? You with me? And how much you squirt out per beat. Yeah? Really simple math, right? So if you want to manipulate cardiac output, how much you squirt out per unit time, you can squirt more frequently, yeah? You can beat more frequently, or you could increase the amount per squirt, right? Or you could do both, right? Exercise, we will see, actually does both. Increases your cardiac output by 2 mechanisms. Okay, so you guys understand this concept before I go any further. Everybody got the concept of cardiac output, okay? So cardiac output is going to what? Positively or negatively correlate to blood flow? Positively, right? As cardiac output increases, pressure tends to increase and flow tends to increase, right? Okay, so what we are going to do is we're going to look at the factors that impact stroke volume, which is how much per squirt, and we're going to look at the factors that impact heart rate, which is going to be easy because it's mostly autonomic nervous system that you already know, right? Okay, so let's do the hard one first. Okay, let's do stroke volume. Okay, so an example, by the way, an adult example of a reasonable cardiac output would be this. Stroke volume of 0.07 liters per beat, which is about like around a quarter cup, and a heart rate of 72 beats per minute, which is among normal, like within normal range, right? Gives you, and this is pretty damn impressive, that cardiac output at rest, at a resting heart rate, is about 5 liters per minute, which is impressive because how much blood do you have? No, you have about 4 to 5. These two big dudes probably have about 5 to 6 liters, between 5 and 6, right? So your entire blood volume is moved through your heart at rest every minute. Every single freaking minute. So maybe you should give it less In-N-Out burger. And a little bit of exercise on occasion, right? You're like, hell no. More cheese. Okay. So now each of these factors is relatively complicated. So what we're going to do is break down this one and then break down this one. Good? Okay. So controlling cardiac output. You can manipulate cardiac output with stroke volume, or you can manipulate it with heart rate. And this should sound relatively logical to you. So I want, let's talk about like an example in which you've actually experienced this. So if I had a heart attack, and I damaged the muscle of my heart, which one of these would be immediately compromised? This one. Yeah? Stroke volume, right? It wouldn't be able to contract very well. But if I needed to maintain my cardiac output because I'm serving the god of flow and I dropped my stroke volume, what would happen to my heart rate to compensate? It would go up, yes. Exactly. That makes sense. Or I have an example that's not a heart attack. It's a relatively benign, usually, unless it happens when I'm driving, kind of cardiovascular condition. And with my cardiovascular condition, I have atrial fibrillation. And what atrial fibrillation does is the atria spaz out and they don't squish all the blood into the ventricles, right? It's just like spazzing out. Which one is this going to immediately impact? Nope. If I'm not squishing the blood into the ventricles, then I can't squirt very much out. So it's going to immediately impact this one. And then what does my heart do to compensate? And my heart rate goes crazy, right? And then I pass out. Sometimes in class. It's happened in this room before, but not very frequently. Only if I'm super duper stressed out does it get really, really massive. Okay, so what we're going to do is we're going to look at stroke volume. You have to pay attention to the stroke volume stuff because this is the part that's probably the hardest for people. However, it's a bunch of little logical relationships. If you get each one, you can get the big picture, okay? So the factors that are impacting stroke volume, I want you guys to brainstorm for a minute. Stroke volume is the amount of blood that leaves per ventricular squirt. Okay, you guys spitball for me and see if you can think of anything that would impact stroke volume. Blood volume, definitely. Okay, blood volume where? Blood volume in the system or blood volume in the heart? Yeah, the amount of blood that's in the heart would impact how much leaves per squirt. What else? Yeah, filling time. Awesome. I didn't expect anybody to come up with that one. What else? I could do or I could go Yeah, how hard do you contract? Yes, all of those things. And then one other thing. Does it intuitively make sense to you that what's going on in the tube that you're pushing into also matters? What if the tube is like this? Yeah? Right, the pressure that you're pushing against also matters. So what's going on here matters, but what's going on there matters as well. So let's look at those. So the factors that are impacting stroke volume. There's a figure in your textbook that shows you this, and we will look at it several times, but we're also going to build it here. The factors impacting stroke volume are three different things. This one is what's happening in your arteries. This one is what's happening in your heart, and this one is what your nervous system tells your heart to do. So let's talk about each of the three of them. This one's really easy. The term is a little confusing, but the concept is really easy. So the first thing and the easiest one to understand for me is what leaves your heart when their ventricles contract has to do with how much pressure they're pushing against. Because this is my P1, right? If my P2 is almost as high a pressure, then I don't end up getting much flow. Yes? Okay. So the afterload is the arterial pressure that you're pushing against. And it could increase really dramatically if I have massive vasoconstriction or arteriosclerosis. Yeah? And what would happen is it would drop my stroke volume. So let's talk about somebody with hypertension, with chronic hypertension, right? With chronic hypertension, your vessels are all gunked up and maybe vasoconstricted, right? And what happens there is I'm trying to get the blood out, but I've got such a massive afterload that I'm working against that it's really dropping my stroke volume. And if I can't do it by stroke volume, how am I going to compensate with hypertension? Your heart rate's going to go up. Yeah? Okay? So afterload is the arterial pressure against which the ventricles are pumping. It's what's going on in your arteries. Increased arterial pressure tends to do what to stroke volume? Decrease stroke volume. You with me? Decrease stroke volume. And afterload increases and stroke volume decreases with hypertension, right? So hypertension first impacts this, and then you try to compensate by increasing this. Yeah? To maintain this, to maintain that, to maintain that. Yeah? That's what you're trying to do. It's all like you're serving the god of flow, right? And if you can't get it by one mechanism, you're definitely going to get it by the other mechanism. Okay, so afterload. So in this figure, you can see that afterload is red. It negatively impacts stroke volume. Do you see that? Okay. Afterload is directly related to what's happening in your arteries. Okay. And it's called after because it's after the heart. We will have preload, and preload's going to be related to what's going on in the heart. Okay. Okay. Questions? You guys are quiet, and it's scaring me. That was the easiest part of stroke volume. Okay.
So you decrease stroke volume, increase heart rate to maintain your CO.
To maintain your cardiac output, to maintain your pressure, to maintain your flow. I mean, not that I would ever ask you in that way, but go ahead.
So when you give CPR, what you're trying to do is like make the flow again go.
Yeah. Uh-huh.
And the SA node will, or the electrical part of it will just start on itself.
We hope. If it doesn't, all you can really do, you can do this, and sometimes the mechanical stimulation of the heart makes the heart pick up the rhythm that you're trying to do. If that doesn't work, what do you have to do? What do you have to do, guys? You have to cardiovert by either, there's chemical means to cardiovert. You can like inject something in them. And what cardioversion does, by the way, guys, it doesn't restart your heart. It stops it from misbehaving. And then you're like sitting there going, you remember how to behave in public, right? And sometimes it doesn't. And then you just die. But you were going to die anyway. You were totally going to die if they didn't cardiovert you, right?
It's so awesome. Just
Yeah.
like last week we had a patient, we had a cardiovert, and that's like really.
Oh, and you work in labor and delivery, huh?
Yeah. It was awesome.
I don't know if you guys know this, but Mike, do women die
Yeah.
giving child? Use some birth control. That's all I have to say, because you can freaking die. What? No, it's true. It's true. I'm sorry, but it is absolutely true. I've known 4 women over the last 10 years who died in childbirth in their 30s. So it's not as safe as everyone thinks it is. And you're not going to die. You're great. But everyone all over Utah, it's just like, have all the babies. And I'm like, come on, guys. You have to realize that this is a risky proposition. It's something you need to undertake very cerebrally. You need to like figure out what's going on. So did she live?
Yes.
Good. I'm glad. Okay. So this is afterload. So afterload, guys. So this is afterload. Afterload decreases stroke volume. And now we're going to get to the hard part. Okay. Put your seatbelts on. It's just really related, and all of the things are kind of close to one another. And this is this concept. This concept is EDV. But before I start telling you about it, I want to break down the term. You ready? End diastolic ventricular volume. End diastolic ventricular volume. Okay. So at the end of diastole is when? The end of relaxation, which is right before systole, which is when the heart should be really, really full or really, really empty, the ventricles. Really, really full. Yeah? Okay. So does it make sense to you that at the end diastolic ventricular volume is directly correlated to stroke volume? How much blood and how full the ventricles are before contraction is going to directly correlate to stroke volume. Yes? Okay, stroke volume. Okay, so this is end diastolic ventricular volume, but it's not just the amount of fluid, guys, because it's a stretchy bag. So it can get full, but it can also get fuller. Yeah? So it's not just the amount of fluid, it's the amount of fluid and how stretched the container is. Okay? Because this is not just like a solid container, it's a stretchy container. So your ventricles can get full, but they can get fuller. Yeah? Okay, so that's end diastolic ventricular volume. Do you have the concept? End diastolic ventricular volume? Okay, so stroke volume is affected dramatically by EDV. This is the volume of the ventricles just before contraction. So let's go through it sequentially, figure out the parts and break it down. You guys tell me. Increased EDV tends to do what to stroke volume? Increase stroke volume, yes? Okay, so let's talk about that. Factors that impact EDV. Okay, so preload is a concept that we're about to talk about. Preload has to do with what's going on in the atria and how much blood is being delivered back to the heart. So let's talk about the concept of preload. The concept of preload, as preload increases, stroke volume increases. And preload is impacted by what Scott came up with, and I didn't expect anybody to come up with. So preload is impacted by how much time you give your heart to fill, right? So if your heart beats so fast that it can't fill, right, then your volume's going to drop and your stroke volume's going to drop. So is there a sweet spot where stroke volume is high and heart rate can be high? Yeah, but if you get heart rate too high, your stroke volume is going to suffer, right? So filling time, which is dependent on heart rate. So as heart rate decreases, filling time and preload both do what? Increase, yes? Now, is there such a thing as too low a heart rate? Well, of course, yeah. 0 is definitely too low, okay? So there's such a thing as too low a heart rate. So filling time impacts preload, okay? If you give it more time to fill, it'll be more full, yeah? And you'll get a greater EDV, end diastolic ventricular volume. Okay, the other thing that impacts it is atrial pressure. So what's in the ventricles depends on what was in the atrium. Yeah? Okay, so atrial pressure has to do with two things. How much is returned to the atrium via what we call venous return. I'll explain that in just a second. And then also how much the atria squish the blood out of the atrium. Okay, so I've got to get it back into the atrium, and then I've got to squish it down to the ventricles, right? Okay, so let's talk about venous return first. Do you know the concept of venous return? Okay, the concept of venous return, some of you know it and some of you don't. It'll make sense once I explain it to you. So we know that blood moves through the arterial system primarily because of the pressure generated by the heart, primarily. But do you know that the pressure generated by the heart is not the primary consideration with blood coming back to the heart? Do you know that? The primary consideration with blood coming back to the heart via venous return, and we're going to talk about this again in just a second, is two things. One of them is the skeletal muscle pump. You guys remember? Okay, so you remember that you don't really have any pulse pressure in your veins, right? So although the heart is maintaining pressure in the entire system, it's really not squirting blood through your veins. So my question is, if blood is relatively low and not very squirty in your veins, how do you get it to move all the way back up to your heart? So when your skeletal muscle squishes on vessels that have one-way valves in them, then you start squirting it up. Yeah? Okay, so that means that with exercise, both you are increasing your heart rate to do the front end, but you're also increasing your skeletal muscle contraction to do the back end. That's called the skeletal muscle pump. Good? But have you also heard of the respiratory pump? Respiratory pump is even cooler. Okay. The respiratory pump is also aiding in venous return. The respiratory pump goes like this. So you've got a whole ventral body cavity, thoracic and abdominal pelvic, yes. Okay, and your diaphragm is shaped like this, like a bell-shaped muscle when it's relaxed. And when you inhale, what it does is it pushes down, and it increases the pressure down here and comparatively decreases the pressure up here, and it squirts blood up your inferior vena cava, right? So every single time you breathe, you aid in venous return. Every time you inhale, you aid in venous return. For that reason, pretty damn fascinating. You have no valves in any of the veins in your ventral body cavity because you don't need them, even though some of them are ginormous, right? So you don't have any valves at all because you breathe pretty damn dependently, right? You're like, I breathe all the time, right? So every single time you breathe in, it goes squirt, squirt, squirt. Just go ahead.
Would that be why you just breathe heavier if your heart rate increases, or if you need to like return blood quicker? Or
There's a whole bunch of different reasons. You breathe heavier because gas changes in your blood. But yes, it definitely helps with this. So let's talk about exercise. So with that concept, we know that exercise does this, right? Sympathetic nervous system increases heart rate. But now we see that exercise actually does this too. It increases stroke volume because of this part. So it increases stroke volume because it actually is going to increase the venous return going to the atrium via the skeletal muscle pump and the respiratory pump increase venous return, which increase atrial pressure, which increase preload, which increase EDV. Got it? Okay, and then we're going to talk about this one in just a second. Okay? All right. So what is EDV? Somebody tell me in your words. EDV. Yeah, this is why this is like, you have to slow down on this because people. No, it's the amount of blood that's in the ventricles, the volume of blood that's in the ventricles, including the size of the chamber, right? Right before.
Increases your output, right?
It increases your output. It's going to increase your output. It's going to increase your stroke volume. Does that make sense? Okay. So, and that is impacted again by filling time. Yeah, lower filling time gives you more time to build up blood in there. And it's also impacted by all of the stuff that is going to your atrium.
So how do the articles impact that?
It's simply like when you get a whole bunch of venous return, instead of the atria getting just full, the auricles can open up and let the atrium get fuller, which increases atrial pressure, which increases preload, which increases EDV. Okay, so let's talk about my problem again. And I want you guys to figure out where the hell my problem is here. Okay, so I have atrial fibrillation. Yeah? It's intermittent. It's related to stress with me. It's probably because I had rheumatic fever when I was a kid. Okay, so I'm going to go ahead and tell you that it massively compromises my stroke volume when it occurs. I want you guys to help me figure out exactly where the problem is occurring in this story. Where is it? Where is the issue in here? It's before EDV, though. It is EDV, but it happens before that. Track it back a little bit and tell me where the problem is. No, it's not filling time. It's atrial pressure. Yes. What are my atria not doing? They're not going squish. They're going spaz, right? And since the atrial pressure drops, I'm not actually doing like coordinated atrial contraction, then I drop preload, I drop EDV, I drop stroke volume, right? My blood pressure massively drops and my heart rate tries to compensate. Does that make sense? Because I'm not doing, I know, what's your atria do, by the way, with atrial fibrillation? Here's what your heart should be doing. You should be going atrial contraction, pause, ventricular contraction. Atrial contraction, pause, ventricular contraction, right? But I'm too uncoordinated to do this. But imagine that this is still going, because I can't do it. But the top ones are just like, they're never like squishing it out, right? And so I'm compromising atrial pressure, and that's the issue there, right? And you can totally feel it, right? It's like, and then my blood pressure really drops. Does that make sense to you guys? Okay. No? Yes? No? You guys are not talking today, except Anna.
So
Go ahead.
When you're saying the atrial pressure, you're talking about that specifically the afterload section of this?
No, afterload's over here.
Okay, so.
Atrial, not arterial. Atrial.
Oh.
Yeah. Atrial pressure. Yeah, atrium is in the atria. So this is atrial pressure. What's in the atrium? Both how much blood and how strong the atria contract affect what's in the ventricles. So that's what this is saying. Not arterial, atrial.
Okay.
Got it?
Yeah.
Okay. All right. So questions. Go ahead.
So EDV is ventricle. So
EDV is ventricle. So what's in the ventricles is directly related to what happened in the atria. Yes? Got it? Okay. Did it click in there? Okay. One other little tidbit about this, and it's related, but it's related in sort of, it goes this way and that way. So here's the cool thing. It's kind of interesting. It's called Starling's Law. Starling's Law says that cardiac muscle sarcomeres contract better when they're stretched. So I'll show you the figure from your textbook. So here's a cardiac muscle sarcomere right here, right? And when it's sort of not very stretched, it actually doesn't contract as well as when it is stretched. So an increase in EDV, if you fill it and stretch it more, it's actually going to contract better. And what this does is it serves to match the output to the inflow, right? So more blood in means an automatically stronger contraction. And the reason, by the way, is that you have more access to cross-bridge cycling when it's a little stretched than you do when it's like this, right? So does it make sense that this might have a greater contractile ability than this one? Yeah? So this is the heart when it's not very full, right? This is the heart when it's really full. And this is not nervous, it's not hormonal, it's just mechanical, right? You stretch it, it contracts better. So this is called Starling's Law. Go ahead.
would exercise have a factor in that? Like
Definitely.
Increase.
Yeah. As you deliver more blood back to the heart, it's fuller, it actually contracts better. And what this does is, as long as this is working, it prevents the enlargement of the heart because you always match output to inflow. More blood in, more blood out. More blood in, more blood out. So this is Starling's Law. So let's look at Starling's Law in your notes. Starling's Law says that as venous return changes, the heart automatically adjusts its output to match the inflow, prevents enlargement of the heart. Basically, cardiac muscle sarcomeres have an optimum length when they're stretched. They work better when they're stretched. They contract better. Increased venous return, like during exercise, will give more blood in, stretch the muscle fibers, and then you're going to get increased output, right? And this is, of course, directly related to the skeletal muscle pump and the respiratory pump. So let's look at the concept map. You ready? Okay, so now do you see why the skeletal muscle pump and the respiratory pump impact venous return? That's what we said. But venous return directly impacts Starling's law, which actually makes EDV work better, right? So these two are related to one another. Venous return impacts atrial pressure. That's what's happening in the atrium. But then it also makes the ventricle work better. Yeah? That makes sense? So this is Starling's law. Starling's law is the length-tension relationship in cardiac muscle sarcomeres. That's what it is. If you stretch them, they work better. And it's just a mechanics thing. So let's reinforce that. Basically, stretching is going to cause the force of contraction to increase. And we haven't even done anything with the nervous system at all yet. We can, but we haven't. So more blood is going to be ejected. Thus, exercise, guys, exercise. We know it increases this one, but it increases stroke volume, yes? And therefore, exercise increases cardiac output. Because exercise not only increases what's happening on the front end, that's heart rate, but also increases what's happening on the back end, which increases stroke volume, right? Because of the respiratory pump and the skeletal muscle pump coming back, right? So now, there is a maximum amount that your cardiac output can increase above normal. So if we have that example that we looked at just a minute ago, in which we've got 72 beats per minute, and it's 5 liters per minute, that's what the normal person. Now I want you to think about Lance Armstrong. Lance Armstrong, at his prime, was just an absolute freak of nature. His stroke volume, why does this always get turned on? Children. His stroke volume was high enough that his heart rate could be 37 beats per minute at rest. Yes, his stroke volume was about 2.5 times the stroke volume of a normal person. And so what happened with him is his stroke volume was so high because of 2 things. First off, when you do cardiovascular exercise, you open up your arteries and arterioles and you decrease what? You decrease blood pressure. What was the thing on the?
No.
What's that called? You're gonna decrease what? Afterload. Yeah? You're gonna decrease afterload, which will increase your stroke volume. But then in addition, his cardiac muscle is really trained, so with every contraction gets massive amounts of blood out. And so he's got that at rest. He's just sitting around pumping like his entire blood volume at 37 beats per minute at rest. But his cardiac output says that just like you, his heart rate can go up to almost 200 beats per minute, right? So he's got this massive cardiac reserve because his resting heart rate is so low. We've all got a cardiac reserve, but if you start from 95 beats per minute at rest, you've got less room to move up than if you start at 37, right? So cardiac reserve is the amount that your cardiac output can increase above resting, and that increases with training, specifically with cardiovascular training. You can increase that. Go ahead.
Like endurance.
Yeah, endurance training, exactly. Yeah. Does that make sense to you guys? Okay, so cardiac reserve is what this is doing at rest and then how much you can increase above that. Okay? All right, so one last thing that determines stroke volume, and it's called ventricular contractility. Okay, so with ventricular contractility, this is something that you may have never been taught before. I didn't run into it until I got to physiology. Okay, so ventricular contractility is this concept that your parasympathetic nervous system, sympathetic nervous system. So let's just figure out what we're looking at really quickly here before we get into the details. So this is the sympathetic nervous system, this is the parasympathetic nervous system going to the SA node and the AV node via the vagus nerve, right? This is the sympathetic nervous system going to the SA node and the AV node via the thoracic region and spinal cord. You guys with me? So this is all autonomic, right? And but what you may have not been told before is that the sympathetic nervous system also directly stimulates the ventricular myocardium itself and makes it contract not just faster, which we've known about for a long time, but harder. Okay, this is called ventricular contractility, and I need to separate it from EDV. So here's what ventricular contractility is. Ventricular contractility says, if I hold EDV constant, I'm not changing the volume, right? because that would be changing two variables at one time. Hold the volume constant, stimulate the wall of the ventricle with the sympathetic nervous system, and I will increase the amount of blood out because the sympathetic nervous system can make it contract harder. Yeah? Now, do you think I could do both of these at the same time? Yeah, right? But here I'm talking about the sympathetic nervous system just making the wall contract harder, and that's nervous, right? Okay, so that's called ventricular contractility, and it's controlled by the autonomic nervous system, specifically the sympathetic nervous system. So again, at a given EDV, don't change the volume. Stroke volume can increase as force of ventricular contraction increases. How do you do this? Sympathetic nervous system either releasing sympathetic norepinephrine, yeah, or epinephrine via the hormone, because it doesn't matter how it got there, right? Okay, and this is why, by the way, epi, yes, you can deliver epi clinically. And what epi does is, for instance, let's say somebody's going into anaphylactic shock because they ate some peanuts or something. It's going to massively, massively drop their blood pressure. And we're going to try to counteract that. We're going to try to counteract that by delivering epi, right? And epi, we know, is going to increase the heart rate, yes, but now I'm telling you it's also going to increase the stroke volume. Yeah? It's going to increase the stroke volume by directly causing the wall of the ventricle to contract harder. Okay? So ventricular contractility is different than EDV because it doesn't change the volume that is in the ventricle. Go ahead, Sarah.
So how does, so in Starling's law, right? It
Yes.
talks about the sarcomeres being stretched.
Yeah.
And then that gives you greater. So is this pulling in faster?
Yeah, it's pulling in more massively, right? So it's starting from, let's look at the Starling's law picture. Where is Starling's law? Where the hell are you? Okay, so imagine that we were here, right, and we didn't change the volume at all. Deliver massive amounts of calcium really quickly and get a really strong contraction in the wall, right? But if you did both of them at the same time, if you also had stretched sarcomeres and delivered massive amounts of calcium, you could get a really strong contraction. Does that make sense? So it depolarizes those cells at the ventricular myocardium. Okay? Everybody good? Go ahead.
So the difference between ventricle contractility and EDV is not does not change the volume.
Yes, exactly. Yes. This is holding volume constant and just making the sarcomeres at whatever site, at whatever size they're sitting, contract harder. It's primarily because of calcium release. Okay? All right. So this is the hard part. Okay? So what I'm going to do over this is I'm going to write some questions that make you kind of pick it apart, right? And then you can talk to each other, and then if you don't get it, then you can come back on Tuesday and we can fix it. Right? We'll fix it. We'll make sure you get them right before you actually. So again, what's today? Thursday? Okay. So I will make the quiz not due before you get to class. I'll make it due the day after, right? And then the answers will populate. Okay. All right. So that is stroke volume. Now the next part. So we already said this. So if stroke volume falls dangerously low, if I really massively compromise this, like someone who just had a heart attack or someone who's bleeding out, then again, just reinforce what is the way in which your body would try to maintain cardiac output. Yeah, it would bump up the heart rate, right? Okay, so these two are intimately related to one another. If one fails you, you'll try to compensate with the other, right? Okay, so now this is going to be easier. The factors that are impacting heart rate are easier because it's primarily the autonomic nervous system. So this is not going to be bad for you guys. So you guys know that the autonomic nervous system is going to control heart rate. And you know, of course, that the autonomic nervous system controls heart rate via medulla oblongata is where it starts. Yes? Yes? Okay. Medulla oblongata is primarily controlling heart rate. Okay. There are a couple of little gray matter chunks in the medulla oblongata. One of them is hooked up to the parasympathetic nervous system via the vagus nerve. Everybody with me? Okay. And this one is going to release what neurotransmitter, guys? Acetylcholine. And acetylcholine is going to do exactly what we just said. Those pacemaker cells were on their way. We slightly hyperpolarize them and slow them down, right? That's how you're going to control heart rate with the parasympathetic nervous system. How you are going to control heart rate with the sympathetic nervous system is use a little different chunk of gray matter in the medulla oblongata. This time, don't use the vagus nerve. Go all the way down to the thoracic region of the spinal cord, out to the ganglia, yes? And then you are going to go up to the SA node, also the AV node, but the SA node primarily. And instead of trying to hyperpolarize it, you're going to depolarize it, and you're going to get a stronger, more frequent contraction. And this mechanism is primarily by releasing which neurotransmitter? Norepinephrine as a neurotransmitter, epinephrine as a hormone. Everybody good? Okay, go ahead.
Does that affect, like, I'm just thinking, like, back to the feet, doesn't the SA node, like, when that malfunctions, doesn't it kind of mess up the, like, filling, or?
Does it? Oh yeah, it totally does. Totally messes up. So right now we're talking about normal physiology. With ventricular fibrillation, that's like pathophysiology. But with normal physiology, sympathetic nervous system is going to increase it. If you're healthy, it's not going to increase it so much that you shut the whole damn thing down, right? It's going to stop once you get into the sweet spot, and it's going to say, I could increase your heart rate more, but that would compromise the system, so we're not going to do that. But in patho situations, you can definitely get into that. Okay, so basically the autonomic nervous system control of heart rate is medulla oblongata hooked up to parasympathetic, okay, via the vagus nerve. That portion of the medulla oblongata that controls parasympathetic nervous system is sometimes called the cardioinhibitory center. Does that make sense to you? Cardioinhibitory center. So, and it secretes acetylcholine, which binds to what kind of receptors, guys? Cholinergic receptors. Yeah, it binds to cholinergic receptors. And it decreases the heart rate. But again, parasympathetic, look, it doesn't have any hookup to the wall, right? So the parasympathetic nervous system impacts heart rate directly and doesn't have any direct impact on stroke volume. The sympathetic nervous system, this is sometimes called the cardioacceleratory center in the medulla oblongata, right? And it hits heart rate with norepinephrine. What kind of receptors? Adrenergic, but specifically on the heart, they're beta adrenergic, which is why we're going to talk about beta blockers on Tuesday. Yeah? Okay. Beta adrenergic receptors, but it also, of course, goes to the wall of the heart. So the sympathetic nervous system, parasympathetic nervous system only directly impacts heart rate. Sympathetic nervous system impacts both. Yes? Okay, heart rate and stroke volume with ventricular contractility. Okay, so now that's why administering epinephrine in a clinical situation has a massive impact on cardiac output and then a massive impact on blood pressure, right? And it's pretty damn effective. Okay, so why is your heart rate not 100 beats per minute if it depolarizes 100 times per minute? It's because the parasympathetic nervous system is consistently keeping its foot on the brakes for your heart. So what's happening right now to you is you're releasing acetylcholine at your SA node, and you're slowing it down so that it's like 60, 70-ish beats per minute. Yeah? So 60, 70 beats per minute. So that means that if you cut all innervation to the heart rate would go up to 100 beats per minute. And interestingly, I didn't know this until one of my students who worked for a cardiac surgeon told me, when they do a heart transplant, they don't even try to hook up the nerves, right? They just like stick it back in there, and they're like, well, apparently it's going to go 100 beats per minute unless we also attach a pacemaker, which quite often they do, because it's just going to go, right? It's just going to like go. And you know that the nerves don't heal very well anyway, and they take 2 years or so to heal. They just stick it in there, and they're like, all the tubes are hooked up. Let's hope you don't reject it. And then, I don't know, maybe it worked great. So the autonomic nervous system regulation of heart rate. So we're going to cover this next time. How do you decide whether to turn on the sympathetic or the parasympathetic nervous system? Well, we said, okay, tigers and no tigers, but of course it's not just tigers, right? It's of course exercise, and your cerebral cortex can do it, but also you're constantly monitoring your own blood pressure to figure out whether you need to turn one on or the other on. And you're also constantly monitoring the chemical content of your blood. Like if the oxygen falls in your systemic arteries, you're going to increase your heart rate, right? Or if the carbon dioxide rises, you're going to increase your heart rate. So there's a bunch of different ways to do that. Okay, so hormonal control of heart rate. So we just did nervous system control of heart rate, which is the same story you expected it to be. It was autonomic. Hormonal control of heart rate is pretty easy too, because epi is a hormone, yeah? And as a hormone, it's released from where? You know. No. Adrenal medulla. And it increases both heart rate and strength of contraction. But then thyroid hormone, T3 and T4, those increase heart rate too, but not like dramatically. All I'm saying is if you're hyperthyroid and it's not treated, you're probably going to have a slightly higher heart rate because it increases the metabolism of all tissues, including your heart. Is that okay? Okay. And then other factors influence heart rate as well. So you know this, you've been a human for a while. If you are emotional, it's going to impact your heart rate, right? Like teaching you cardiovascular physiology at the end of the semester. You're like, you are stressing me the hell out, Melanie. You can see I can't teach this until the end of the semester, though. Sorry, guys, I just can't. You don't have the background for it. So emotions can do it. But then temperature can also do it. Since it's all based on diffusion, right? Higher temperature, faster diffusion, faster heart rate. Lower temperature, slower diffusion, slower heart rate. But luckily, lower temperature, slower heart rate is fine because it also lowers your metabolic needs, your oxygen needs, okay? And then endurance training, like Mike said, endurance training, what it serves to do is to increase your stroke volume so that your heart rate can decrease, right? That impacts long term. Okay, last thing for today is to talk about when things go bad, okay? So congestive heart failure. With congestive heart failure, here's what I want you to think about for just a second. Okay, if your ventricles go bad on you, like you have massive hypertension and your ventricles can't generate enough pressure to overcome it, or if you just had a heart attack and your ventricles can't generate enough pressure, right? What it's going to do is it's going to really massively impact the blood that can leave the heart, but not so massively impact the blood that can come back to the heart. Because remember, the blood that's coming back to the heart is more related to venous return via skeletal muscle pump and respiratory pump. So what will happen is you're going to get a traffic jam. You're going to get congestion. Okay? Congestive heart failure is when one of your ventricles, maybe both, but let's just say one at a time, fails you. And what you're going to have to do is with congestive heart failure, the input keeps coming in, yeah, but the output can't keep up with it. And you're going to back up until you hit a capillary bed, and then you're going to leak fluid out the capillary bed. Okay, so let's see what would happen if you had failure of the left heart, failure of the left ventricle. So I had a heart attack and my left ventricle is really weak. Okay, where am I going to start leaking fluid? Back up. Okay, comes from the left ventricle, came from the atrium, right? Traffic jam. Backs all the way up to where? Backs all the way up to the lungs. And you get what's called pulmonary edema or pulmonary pleural effusion, excess fluid around the lungs. Do you know that shortness of breath is typically associated with cardiovascular disorders? This is one of the many reasons, especially when you're laying down. So go ahead.
Pneumonia, part of that?
It can lead to pneumonia. The fluid can leak into the actual tissue of the lungs and cause pneumonia, but this is usually around the lungs. It like leaks into around the lungs. Okay, so this can cause shortness of breath, but what if you had failure instead of the left heart, you had failure of the right heart? I want you to track this back. Failure of the right heart is going to track back to which capillaries? Systemic capillaries, but now add gravity and tell me where I'm going to get a lot of fluid buildup. Hands and feet. Okay, so if you wake up with swollen hands and feet and you're not pregnant and you didn't eat a huge bag of Doritos yesterday, right? Then it's not a good sign if you wake up with that every day. What it's going to do is it's going to cause edema. You're going to have swelling of the ankles and the legs, sometimes of the hand as well. You might even get ascites. And that generally means that what's happening is the blood is backing up in your venous system until it gets to the capillaries and it's leaking out of the capillaries into your interstitial fluid. and you're going to get edema of the tissues. This is an indication that something is not happy with your cardiovascular system, and it's called congestive heart failure. And what will happen with congestive heart failure, and then I'll let you guys leave, is it can lead to try to compensate for it, your heart will increase in size. This is the left ventricle that was dealing with long-term congestive heart failure. And what happens is it continues to try to compensate by increasing the size and the strength of the ventricles. Unfortunately, what it does is actually decreases the filling capacity of the ventricles, right? Because there's nowhere for that pressure to go. So that's congestive heart failure. That's what that is. Good. Did you have a question?
I don't even know if this terminology is right, but does the backbone end up like breaking the cord tendon because it's back?
Oh, it totally can. Yeah. Yeah, it can. It's not usually, it's a combination of congestive heart failure and usually hypertension that can cause the breaking of the chordae tendineae. My father-in-law had that, but he had it associated with hypertension, obesity, and then he also took fen-phen. Did you remember the drug fen-phen? Phentermine and fenfluramine, which was a diet drug. So what that did is it calcified the chordae tendineae so that they got kind of brittle, right? And in combination with the other two things, his mitral valve just prolapsed and popped the chordae tendineae. And now he's got a mechanical valve in there.
They're really strong.
Yeah, they're really strong. Yeah. And they don't usually break unless you have other complicating factors. And that drug, fen-phen, the diet drug, massive lawsuits because of it, and he was part of the massive lawsuit. It caused them to calcify and break. It's like when the seals on your car get all dried out from salt and heat and everything. So that happened to his chordae tendineae, and then when they pushed against them, they just popped open. So I'm surprised he's still alive, but he's lived with that mechanical valve for like 15 years now.
So is congestive heart failure kind of similar to ventricular fibrillation, where it can't like push it all out, so does it cause back?
Yeah, but ventricular fibrillation would be an acute problem, and congestive heart failure would be a chronic problem. But they are causing exactly the same thing to occur. Yes, go ahead.
So